Good afternoon, my dear students. Today we will be continuing with chapter number 6, Molecular Basis of Inheritance from class 12. Now, in yesterday's lecture, we have seen about the DNA molecule. Now we will be learning about the model of DNA. The model of DNA, also known as double helix model, was given by James Watson and Francis Crick in the year 1953. One more rule was being given by Chargaff in 1950. This rule states that the ratio of adenine and thymine and cytosine and guanine will always be equal to 1. That means whatever will be the number of base pairs of adenine and thymine and the cytosine and guanine will be equal to unity, or the ratio will be always constant. This was Chargaff's rule. Now we will be learning about how Watson and Crick have deduced this model. They have taken help from Maurice Wilkins and Rosalind Franklin, and they have produced the X-ray diffraction which has produced this structure, and this structure is known as the double helix model. In this we have got two DNA strands with anti-parallel. What does this mean? The one strand will be 5' to 3', and the other strand will be 3' to 5'. So these strands are basically the phosphate group, and what we have got inside, we have got the sugars which protrude inwards, okay, and we have got nitrogenous bases like cytosine will be matching with the guanine and adenine will be matching with the thymine. So adenine will be matching with 2 hydrogen bonds with thymine always, and cytosine will be matching with 3 hydrogen bonds pairing up with guanine. They can never be reversed. That means always a purine will be matching with the pyrimidine. So that means the plane of the base pair will be stacked together to give the stability to this double helix model of the DNA. Now we'll be coming to the pitch. The pitch means the pitch of the helix will be pitch means, suppose, have you seen the spring? The spring goes like this. So the one height of that rounded spring will be 3.4 nanometer. And in one turn of the double stranded DNA molecule, it will be having 10 base pairs. That means the distance between the two base pairs will be 3.4 multiplied by 10 will be giving me 0.34 nanometer. That means these hydrogen bonds will be giving stability to the DNA molecule. So inside will be the sugar molecule, then the nitrogenous base, here then again a nitrogenous base, then a sugar molecule. So like this way, two parallel DNA strand, anti-parallel DNA strand will give the stability to the DNA molecule. And in the E. coli, the length of the E. coli is 1.36 nanometer, and the length of DNA helix in humans, will be approximately 2.2 meter. How this calculation came, we will be learning in the next lecture once we will be dealing with the packaging of the DNA helix. Because as you know, these phosphate bonds are negative in charge. So there must be some positive entity to give more stability to this DNA molecule. That will be the histone protein. We will be learning more about the histone protein. Right now, in terms of your board exam, Watson and Crick model is very important for your long answer. Please students, go through this diagram, go through the laws, go through the years which are mentioned along with the name of the scientist, and please go through these calculations, and proper diagrammatic explanation is there given in your NCERT textbook. Please do read this from your NCERT, and we will be solving obviously all the queries out there. Hope so you all are studying and staying safe at home. Thank you, students.